Here we have our DNA strand, and we begin DNA replication with helicase at the origin. Helicase is inside the DNA strands, and here we have the replication fork. As helicase is moving down the strands, it is unwinding them and spreading them apart. When this happens, it causes tension, which will then lead to supercoiling and lead to DNA replication to stop. To prevent this from happening, topoisomerase is on the outside of the two strands to prevent that from happening. As helicase is going down the two strands, SSBPs are attaching to the two strands to make sure that they don't bind back together. And for this video, we'll be only working on one end of the strand rather than both ends. Just note the DNA replication happens on both ends. For this video, we'll be working on this side of the strand. So as helicase is going down the strand, and SSBPs are binded to the two strands, primase is going down the strand and making RNA primers on both ends. That way, DNA polymerase can go ahead and start to make the contemporary strand from the template strand. The two strands are anti-parallel, meaning they are complement of each other. Notice how the 3 to 5 direction is going to the left side on this end. And notice how the 3 to 5 direction is going to the right in this direction. Since this is going to a 3 to 5 direction, this is the leading strand, and this is the lagging strand. The leading strand is long and continuous. The lagging strand is not continuous because it only goes to a 3 to 5 direction. Every time DNA area is exposed, DNA polymerase has to backtrack. And primase has to go back to that area and put down primer continuously for the lagging strand. These fragments of DNA pieces are known as Ozaki fragments. Since the primers were made of RNA, they need to be placed with DNA bases. So, ligase fills the gaps of the Ozaki fragments together. So once they're sealed, now you have two strands. One from the template and one from and a new one, which is a complementary. And over here, we have the template strand and the contemporary strand. And that is how DNA replication happens.